Okay, folks, let me pick up where we left off on our muscle talk that we had on our other video just previous to this. Let me just sort of recap a little bit. We know that what happens is the signal initially for muscle action originates in what we call the motor cortex in the brain. And we'll talk a little bit more about where that motor cortex is, exactly where it's located in the brain when we get to our next section, the neurology section. And what happens is at that point, once those cell bodies are stimulated in that motor cortex, the signal starts to go down an axon, and that's that long string that extends from the cell body, the nerve cell body, that could go many, many feet. What it does, it goes down to the spinal cord and actually crosses in the brain from the, if it's on the right side of the left side of the to the right, goes down the spinal cord and exits, like we talked about last time, out one of those spinal nerves that comes out between those gaps on the sides of the vertebrae. And as a result, then that nerve, which actually has a bundle of a number of different axons or nerve fibers within it, heads towards its final destination. When it finally gets to that final destination and that signal finally gets down to that motor end plate, that area where the end of the nerve comes very close to the sarcolemma, or the muscle cell membrane, with that synaptic gutter and that synaptic cleft like we talked about last time. When that signal finally gets down to that axon, that nerve terminal at the very end of the nerve, it triggers a release of chemicals such as calcium, which actually causes the acetylcholine in those small little vesicles that are at the end of that nerve terminal to be released. That acetylcholine then migrates its way across that synaptic cleft and attaches to receptors, acetylcholine receptors, along the course of the synaptic gutter. And as a result, what that does is that starts, if there's enough of a stimulus, enough of a signal, that stimulates what we call an action potential, which we discussed briefly on our last video. This action potential starts to send this electrical signal along the whole sarcolemma. Okay, and that's where we're at right now. We also should mention before I head on that what will happen at that point as well is to prevent re-excitation by the acetylcholine, that what happens is that cholinesterase, that enzyme like we talked about in our last video, actually deactivates and breaks down the acetylcholine and takes the choline and moves it back into the nerve terminal so more acetylcholine can be made. It's sort of like a, very green, if you want to talk about that way. We're reusing materials, repurposing that choline to make more acetylcholine in the axon terminal that can be used for more muscle contraction. However, then now we have to think about what really happens with that sarcomere itself. As that signal starts to go down the sarcolemma, the membrane, we talked last time that there are various cisternae, or at the ends of the sarcolemma, that contain a lot of things. And a lot of these things, one of the more important things it has in there is calcium. We talked about how the calcium, and I mentioned this in our last video, that calcium we always talk about as being so important in regards to bone strength, but actually we use the calcium a lot more for muscle contraction. What happens is the calcium that's contained in vats, in these cisternae, which are in the sarcolemma, are released. Okay, and that gets us to where we talked about with our actin and myosin. Remember we talked last time, and let me just go to the first slide here, and they'll probably get us back to where we were before. Remember we talked about actin and myosin. Actin is a very thin filament. It goes from one Z band to across to where it interdigitates or overlaps with the myosin. We know that that myosin is then attached in the middle of the sarcomere. So basically that's where it anchors to the center of the cell. On the sides of the myosin, we know there are small little paddle-like structures that will actually sort of grab the actin and move it and pull it, which actually causes an interdigitation and overlapping of the filaments more and more and more. However, that's prevented by the fact that we have troponin and tropomyosin that really sort of block that for the time being. Okay, what happens is the calcium then will actually unloose this. It sort of like is the grease that lets things happen. It stops that. It stops the limitation or the prevention of the actin and tropomyosin from interacting and allows that interaction to occur. And that's where we get what's called the sliding filament theory of Huxley. And what we happen is the way that the muscle contracts and the way that that sarcomere gets shorter is because now the myosin filaments pull the actin filaments by these little paddles towards the center of the sarcomere, and by doing that, it causes a shortening of the length of the overall sarcomere, which then is what we see as muscle contraction. Those Z discs, where the actin is attached to on both ends of the sarcomere, gets closer, brings in closer, so it shortens the sarcomere, and the sum of the shortened sarcomere means that the muscle is shorter. Now, again, what happens is firing of one sarcomere doesn't make a whole lot of difference, but this happens in multiple, many sarcomeres, and that results in a shortening of the muscle. And muscle can actually shorten, if dependent upon the number of sarcomeres that are stimulated, up to almost a quarter of it, almost up to about half of its resting length. So it could shorten quite profoundly with that. And this is what we call that sliding filament theory of Huxley. Let me just try to show you a little picture of what we're talking about. If we look here at this diagram up at the top, okay, let me give my little thing here. And if we look at this diagram up in here, what we see is here's my actin filament. Here's an actin filament here, actin filament here, actin filament here, and stuff like that. And then what happens is we have that actin filament, over here, and here. Here's the Z disc right there. Here's the Z disc, which is the end of the sarcomere. What happens is this area right in here is the myosin. This is the myosin right here, and it's attached right here at that center anchor, that M line that we talked about in our last diagram. What happens is, again, these little paddles on the side of the myosin are prevented from interacting with the actin because of the troponin and the tropomyosin, which is attached to the actin filament. However, when that nerve, that sarcolemma, like I mentioned before, is stimulated, it causes calcium to be released into the sarcoplasm, or in the cytoplasm of the muscle cell, which then breaks the lack of adhesion, or allows the myosin then to interact with the actin. As a result, what happens is the myosin pulls the actin, so this part of the myosin will pull the actin this way, and this part of the myosin will pull this part of the actin this way. And as a result, the distance from one Z band to the other Z band, or Z disc, from here to here will shorten. Okay, and that's what we see with muscle contractions. Here, down here, this would be a partial muscle contraction, where we see a little bit more overlapping of those filaments, and down here is much more severely or significantly maximally contracted, where now the myosin and the actin, are very closely parallel to each other. There's a lot of overlap. As a result, the idea here is to take that Z disc here and that Z disc here, and now it comes closer, and that causes the muscle to get shorter. Okay, and that's basically the basis of this sliding filament theory of Huxley. This is just another diagram that shows the same thing. It probably should have been done the other way. If I look here, this is basically, here's a Z and here's a Z over here, and this actually here is an uncontracted muscle. See, here's the purple would be the myosin filaments, and all those little dots that we see along the purple are those little paddles. Here's the actin filament right here with all the troponin and the tropomyosin sort of intertwined on it, which by being active doesn't allow the interaction between these little paddles and the actin filament. However, when the calcium is released, it allows these little paddles, you can see a little paddle right there, little paddle right there, like there. What they do, it allows them to interact, and it pulls the actin filament. And by pulling the actin filament, it pulls the actin filament this way. So it pulls the actin filament here this way. And as a result, if I look at my Z right here, and I look at my Z right here, they get shorter. They get pulls this way, gets pulled this way, and thereby muscle gets shorter. That sarcomere gets shorter. And that's the whole purpose, or the whole basis, is what happens with this muscle contraction, why the muscle cell is able to contract, that contractility of it. So basically, here's resting, like I mentioned before, where there's overlap, but not a whole lot. And again, we know that my myosin is banded together here at the center of the sarcomere right here. The Z disc is out here. Then if we look up in here, here's my Here's my resting, okay, or down on the bottom, and the top is my contracted. And you can see how much more overlapping there is of the actin and myosin. So here's an overlapping of the actin and myosin, all this area in here, which is a lot more than we see down in here. And that's what makes that whole muscle sarcomere, that muscle cell, get shorter in length, okay? So basically, we know that the neurotransmitter, which is acetylcholine, and I think I would definitely remember that, okay, is released from the end of the nerve into the neuromuscular junction. As a summary again, if there's enough acetylcholine that's put in there, okay, enough of this neurotransmitter to reach that threshold, what happens is the whole muscle cell will contract. It's called an all or none principle, and that's an important principle that we have to talk about. All or none principle. We can't. What it means is that the muscle is either going to contract or it doesn't. The muscle cell either contracts or it doesn't. Not, it's either complete contraction or no contraction at all, okay? The more sarcomeres that are contracted, the shorter the muscle is going to get, okay? So it pertains only to the cells that are innervated and stimulated as to what is contracted, not to the entire muscle. Roddy, remember that what happens is the entire muscle is supplied by a number of nerve fibers, okay? And some of them will overlap in regards to where they originate from. But these number of nerve fibers, basically, each will work independently. So if I need more, quote, recruitment, which is a word that we talked about last time, more of these fibers are going to be stimulated, they're going to stimulate more sarcomeres, and more sarcomeres by the all or none principle are going to contract, and the muscle is going to have more shortening, therefore more power, okay? So that's what we see. The more muscle cells are stimulated sufficiently, the stronger the contraction would be, and that's just what we talked about there, okay? A single nerve conduction produces a contraction, and that would be called a muscle twitch, okay? So basically, that's a little bit of a summary again as to what we have in regards to muscle contraction. And again, this is just another view, looking at this, the fact that we have here's the muscle, a bundle of muscle fibers together. The nerve, here's the nerve coming. The motor end plates would be in this area. The motor end plates would be in here. There's a blown up picture of that motor end plate, and all those little areas in here are the areas with the acetylcholine that are that become released into that synaptic cleft into here, that stimulate and activate and stimulate the receptors on the sarcolemma that causes that action potential to be produced, okay? And again, once I get that action potential, just means enough of that, there's enough stimulation of the acetylcholine for that to happen, okay? Following the stimulation, again, that neurotransmitter, that acetylcholine, to prevent re-stimulation of the nerve, is inactivated by cholinesterase. And that, again, it breaks down the acetylcholine, and finally the choline is transported back into the nerve terminal where it can be used to make more acetylcholine. Once contracted, the muscle cell is unable to contract again for a specific period of time. Now that's important, and that's called the refractory period. What happens is, let's talk and go back to what we talked about a little bit last time. We know that what causes that stimulation along the sarcolemma is because what happens is pores start to open and these ion channels start to open, and that's when my sodium, as we talked about last time, rushes into the cell. To try to balance that out, eventually the potassium is going to rush out, because both of them are cations, or positively charged materials. So therefore, to try to balance that, the potassium rushes out. Now what happens though, after this contraction occurs, if it's significant enough, sodium's on the inside, potassium on the outside, which is not the way it's supposed to be. So we have to actually have some type of a pump, which is more active transport, to be able to transport the sodium back outside the cell and the potassium back to the inside of the cell, which means that there's a period of time where that muscle's not quite right, and that's what we call the refractory period. It's during this refractory period that the muscle can't be re-stimulated. It's a very short period of time, but it's called the refractory period. No further stimulation during this refractory period will result in a contraction. Why? Because I don't have the ability, if the sodium is not on the outside, the potassium is not pumped back to the inside, I can't have that transfer to occur, which actually started what that electrical stimulus would be. I hope you understand what that means. It takes some time to reestablish those gradients again, to get the sodium back to the outside and to get the potassium back to the inside, and that's what results in that refractory period, okay? And that's an important factor to remember as well. So let's just go over this again, because I think it's quite important to be able to understand this and to recognize this. First of all, that neurotransmitter, acetylcholine, let's we all know that how it comes from the brain and goes down to the nerve terminal, and the acetylcholine is released. And we'll talk more about that when we talk about the neurological system as well in about a week. Okay, so let me just start with this. The neurotransmitter, the acetylcholine, after that stimulus reaches the nerve terminal, is released into that neuromuscular junction or that synaptic cleft. This, if it's sufficient enough, causes depolarization of the sarcolemma, and if it causes what's called an action potential. And that action potential is just this electrical signal that is transmitted all the way down that sarcolemma to the entire muscle, the sarcomere itself. What happens, we get down to those T tubules. We talked about that just briefly a few minutes ago, and those T tubules have cisternae in it, and these cisternae are the areas where we have a lot of calcium. Once we stimulate the sarcolemma going down the T tubules, these terminal cisternae, which are next to the T tubules, which are down the ends of the sarcomere, open the calcium gates, allow the calcium to be released into the sarcoplasm. That calcium then binds with the troponin, which allows then the myosin to start to pull on the actin. Okay, it causes the calcium, when it binds the troponin, causes the shape in the tropomyosin troponin, and as a result, that allows the myosin binding sites on the actin to be uncovered, and the myosin then pulls that actin, which shortens from one Z disc to the other Z disc, shortens the distance between the two, and that's a muscle contraction. Okay, hopefully you're able to follow all that, and I could always answer questions if you don't quite understand that, but that's how muscle contraction basically occurs. Okay, if I go through this sort of sequentially, and again, I keep on going over this and harping it because, again, I know that on an examination I'm going to have to ask some questions. Let's just go these sequentially. 1. Nerve impulse arises at the axon terminal of the motor neuron and triggers the release of acetylcholine. We know that. We've talked about that probably 15 times already. And there's the acetylcholine right here being released into that synaptic cleft, and that which is made between the nerve terminal and that synaptic gutter. Acetylcholine diffuses across the synaptic cleft and binds to its receptors on the motor end plate and triggers a muscle action potential. So along that area of that sarcolemma here in that synaptic cleft are acetylcholine receptors. The acetylcholine gets there, causes a receptor, and that causes stimulation. Okay, the acetylcholinesterase in the synaptic cleft destroys acetylcholine, so another muscle action potential does not arise. Okay, unless more acetylcholine is released from the end of the nerve into that synaptic cleft. What this does is it triggers this action of an action potential. Okay, and that action potential is that electrical signal. The muscle action potential travels along the T tubules and opens calcium release channels in the sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum membrane, which allows the calcium ions to flood into the sarcoplasm. So basically, as that action potential goes along the sarcolemma, that muscle cell membrane, it triggers the release from these terminal cisternae, which are these vats that contain the calcium. It triggers the release of the calcium into the sarcoplasm, so now it starts to fill the sarcoplasm. Okay, the calcium binds the troponin on the thin filament or the actin, exposing the binding sites for myosin. So now those little paddles on the myosin can start to pull on the actin as that happens. Contraction power strokes uses ATP, which is energy, which is stored on the tropomyosin, and the myosin heads bind to the actin, and what they do is they keep on grabbing and release. I like pulling hand over a rope, and if you have two people at two ends of a rope and you want to pull one person closer, say you tie the rope around somebody's waist. Here's an example, and I please don't get arrested if you try this, but if you took a rope and tied it around somebody's waist and you put them across the room and you grab that rope at the other end of the room, how do you bring that person to you? You take your one hand, put it forward on the rope, and you pull. Then you take your next hand, your other hand, put it forward in the rope and pull. Then you keep on overlapping your hands and pulling. And that's what the myosin does to the actin. It pulls it like a rope along that whole length, and that's what causes this shortening from one Z disc to the other Z disc, and more overlapping or over-digitization of the actin and myosin filaments. Okay, the calcium release channels in the sarcoplasmic reticulum close, and the calcium actively transports the use active transport pumps use ATP to restore the low level of calcium ions in the sarcoplasm. So that's trying to get the calcium balance back, and the troponin-tropomyosin complex slides back into position where it blocks the myosin. So once this contraction occurs, we have to pump that calcium back into those terminal cisternae, which then requires energy as well. So now the calcium floods in there, it takes away that blockage of the myosin pulling on the actin. That occurs, but then what happens is then with the calcium, when that occurs with the contraction, then the calcium is pumped back into the terminal cisternae. Therefore, now these binding sites become blocked again, and allows the actin and the myosin to slide back to their original length. Okay, and that's what happens with a muscle contraction. Pretty basic. Okay. Muscle contraction. Certainly, the ATP on the myosin supplies energy for activating the actin binding sites between the actin and the myosin, and ATP, which is adenosine triphosphate, releases one of the phosphates, and the release of the phosphate is what actually provides the energy. If you've had basic biology, you realize that what happens is ATP is a high energy compound because it has three phosphates bound to this adenosine. Okay, by releasing of one of the phosphates, it also releases energy. The problem is now the ATP then is converted to ADP, adenosine diphosphate, because it's lost one of those as it's provided the energy. And when the energy is provided, the myosin heads attached to the actin, results in that power stroke, or that pulling of that rope and pulling the actin closer over the myosin. The actin filaments are pulled more towards the center, where the myosin is attached in the center of the sarcomere. The ADP detaches. New ATP binds to the myosin heads, causing the detachment to actin. Myosin then become reactivated. So that's that power stroke, one stroke after another. Okay, and it continues as long as there's high calcium ions in the sarcoplasm. Now we talked just a few minutes ago about what happens is then I have to take that calcium from the sarcoplasm and put it back using energy back into those terminal cisternae. So that what happens is that we can go back to normal, and we can have that lack of the less overlapping of the actin and myosin as the muscle sarcomere gets back into its normal length. Okay, and that's what we've talked about so far here. That's just another reiteration of what we've talked about thus far. Okay, and again, this is just looking at what happens. The myosin heads hydrolyze the ATP, become oriented and stuff like that. As a result, we have the myosin heads bind to the actin and pulls it. Okay, as it pulls it, we have more overlapping of the actin and the myosin, and that's what causes the muscle, the sarcomere, to get shorter, and that's what you have to remember here. Okay, muscle relaxation occurs when the acetylcholine becomes decreased or broken down by the cholinesterase. Okay, the action potential discontinues. In other words, that electrical stimulation of the sarcolemma stops. So as a result, at that point, the muscle contraction will stop. Okay, and again, that's at the point also where the terminal cisternae are reestablished with their levels of calcium, as the calcium gets pumped back into those terminal cisternae and put into those vats where it could be reused again. Okay, don't worry about how it's held in the cisternae, but we'll talk about that's not really important for the level that we need to talk about. And basically, the muscle then goes back to its original shape, because again, the calcium is important because once it gets in the sarcoplasm, it causes a change in the troponin-tropomyosin arrangement around the actin, which allows the myosin to grab it and pull it, as we talked about before, and that's what causes that muscle contraction. And that's pretty much what we talked about. That just shows it again. Okay, muscle relaxation. Again, what happens is after that muscle contracts, we see it here in this contraction period, right, through here, has this relaxation period. And this relaxation period, again, has that period of time where the muscle now goes back to its normal length, where I have a reestablishment of my potassium and sodium gradients on the inside and the outside of the cell, and the calcium is pumped back in those terminal cisternae. Why? So we could do it all over again, just like I've repeated this over and over and over again over the last couple of minutes in regards to how muscle contraction does occur. Okay, a little bit about muscle metabolism. Again, ATP is an absolute requirement. Okay, and that's what you have to remember. That ATP lets things happen. I need energy to be able to make things happen. So the ATP is important. Also, creatine phosphate. Creatine phosphate is also part of that energy system that will provide muscle energy as well. Okay, and what happens is that's why you see muscle builders, one of the supplements they sometimes take is creatine, and the creatine is basically another energy source that we do have for muscle contraction. Glycolysis just means taking the glucose as a nutrient and breaking it down to be able to make ATP. Now, when we talk about aerobic or anaerobic metabolism, okay, which follows this glycolysis, once I take the glucose and start to break it down, it goes through this this cycle where what happens is now the C6H12O6 glucose is actually going to end up being broken down to H2O, water, and CO2, carbon dioxide. But what happens is what in the process of how this happens, it depends upon if I have oxygen present or if I don't have oxygen present. If I have oxygen present, okay, which is what we call aerobic metabolism down in here, I have a very high yield of ATP. Okay, if I have anaerobic metabolism, okay, where there's not much oxygen, what happens is I can't take, when I start to go through this first breaking down of the glucose, I can't make a lot of energy. And this is called the glycolytic lactic acid pathway. And what happens is at this point I have a very low oxygen yield, only like about 2 ATP per unit of glucose, where down here on my aerobic metabolism might get about 30, 36, 38 ATP per unit of glucose. So therefore the importance is I'm able to provide a heck of a lot more energy in high oxygen environments as compared to where I'm a lot less energy in low oxygen environments. Okay, people when they do aerobics, the idea is long, slow, sustained activity teaches the body how to utilize the oxygen a lot better, and as a result what happens is it builds up their ability to be able to transport and to remove oxygen. And why is that important? To make more energy. Okay, as compared to anaerobic metabolism. So the whole thing here is basically we're wanting to really think that the aerobic metabolism is much more important, much more beneficial, because it does have a significantly higher ATP yield. And what I mean by ATP yield, energy yield, higher energy that's produced by the aerobic metabolism. Okay, and that just goes to the little thing. You don't have to worry about that. I won't ask you to reproduce that. Okay, same thing happens here. If it turns out that that the glucose is broken down, and basically with lower levels of oxygen, one of the products that we have here is lactic acid, and lactic acid is basically a byproduct of the breakdown of glucose. And basically in that case, that's what builds up a lot of times in muscle. I take stretching after exercise says sort of like that people will say, well, actually sort of squeeze some of the lactic acid. And what happens, we find people with oxygen debt, the lactic acid levels in the blood go up, which decreases the pH in the blood. The pH in the blood goes down. Okay, and the pH is basically the acidity of the blood. So if the pH is low, it means the blood is more acid. If the pH is high, it means that blood is more basic. So basically this lactic acid is not the way we really want to go, and that's by low energy or low oxygen, low ability to produce ATP. Okay, so in conditions of lack of oxygen or anaerobic conditions with an oxygen debt, lactic acid, like I mentioned, is a byproduct and accumulates, which does cause muscle cramping, muscle soreness, and stuff like that. And we have depleted fuel sources much faster. In other words, if I have 1 unit of glucose that's only making a couple units of ATP, as compared to 1 unit of glucose which is making 36 to 38 ATP, which is the best? Okay, it's obviously going to be using the oxygen source, the aerobic type exercise. So as a result, in anaerobic activities, one of the things that occurs a lot more commonly and much more readily is muscle fatigue. So if we look down here, muscle fatigue is one of those problems that we see with anaerobic type of activity.